Welcome back to Chem 115, Week 11 Recitation. As you know, we're doing our recitations virtually now, and this is the first attempt at trying to do this. This video today will introduce Lab 8, which is how can we use chromatography to separate plant pigments. Some general information. This lab is not in your lab manual, but the lab procedure, pre-lab, and report form are already posted on Blackboard in the Lab 8 folder. This lab is due next Monday, March 30th at 11:59 p.m. Eastern Time, so make sure you upload your pre-lab and report form by that time. Normally we'll have students work in pairs and bring their own leaf samples. But unfortunately, this is not going to happen this semester. So instead, we'll have two TAs do the lab and post their data for you guys. So you use their data to complete your report form. The objectives of this lab, you will learn the technique of TLC and learn how to prepare samples for TLC. So TLC is a type of chromatography. I will introduce chromatography a little bit more in the next few slides. So essentially, using this technique, you will be able to separate the different pigments from the tree leaves and spinach leaves. And each of those components will have a different RF value. So by calculating the RF values and comparing them with the spinach reference, you should be able to identify what those components are as best as you can. Later, we'll try to use the concept of intermolecular forces to explain why those pigments will have different RF values. So chromatography is a very useful technique to separate components of a mixture. For example, our plant leaves have a mixture of different pigments, like the green ones, chlorophylls, the orange ones, carotene, and some other ones. So what if we want to study the pigments individually? We'll have to separate them using the chromatography technique. There are different types of chromatography, like column chromatography, gas chromatography, and thin layer chromatography. So thin layer chromatography is what we're using in this lab. The thin layer is actually a layer of silica gel that's coated on a plastic sheet. Although there are different types of chromatography, the principle behind them are all the same. So components are separated based on their different interactions with the stationary and mobile phase. So for example, for the thin layer chromatography, the stationary phase, which means the phase that's not moving, is the silica gel. And the mobile phase, so the moving phase, is the solvent we're using. So based on the different polarities of our components, the interactions between the components with the mobile and stationary phases are going to be different, which will separate the components. Now we will talk a little bit more about the stationary and mobile phases. We already said that the stationary phase is the silica gel, or silicon dioxide. So looking at the structure here, we see many OH groups on the surface of the silica gel, which makes it very polar. So the silica gel will interact with the polar components in the mixture. For the mobile phase, we have a solvent mixture of acetone and hexane. So for your pre-lab assignment, it asks you to draw the Lewis structures of acetone and hexane. So think about, are they polar or nonpolar? Actually, hexane is just a long alkyl chain. That's just carbon and hydrogen atoms, which means it's very nonpolar. So although acetone is slightly polar, but overall the solvent mixture is much less polar than the silica gel. So when you load your sample on the TLC plate, so your sample here, the mixture will dissolve in the solvent, and the solvent will carry them over and move up. So the nonpolar components will interact with the relatively nonpolar mobile phase stronger. So they'll move very fast and appear on top of the plate, versus the polar components will be attracted to the polar silica gel and will not move up as fast. So there are the slow moving spots right here at the bottom of the plate. This is actually a much nicer picture of a TLC plate than my drawing on the previous slide. So it is a TLC plate of spinach standard. So this picture is from your lab manual showing you all the pigments that are extracted from the spinach sample. So we already said the polar components will not move up as fast, so they will appear here, and beta carotene is the most nonpolar component in the mixture, so it will travel the fastest and be here. So when you have your TLC plate of your plant leaves, you compare your TLC plate with the standard and then try to identify what the pigment components are. This slide gives the chemical structures of some possible components. You don't need to memorize all these structures, but by looking at their structures, you should be able to tell which one is the most polar, which one is the least polar, so compare their polarities. So you might notice that the chlorophyll A and B have very similar structures. So the only difference we see here is chlorophyll A has a nonpolar CH3 group, but chlorophyll B has a polar CHO group here, which makes chlorophyll B more polar than chlorophyll A. What about the xanthophyll and the beta carotene? Their structures are similar as well, but the xanthophyll has a polar OH group right here, which will make it more polar than the beta carotene. So actually, xanthophyll is the most polar component because of the very polar OH group. So it's going to be more polar than the chlorophylls too. But the beta carotene is actually the least polar component here because it contains only the carbon and hydrogen atoms, while the other three have oxygen atoms in there. So looking at the TLC plate here, it should be consistent with what we just talked about. So that's so much for the background information of the lab. Now we will go on to talk about the procedure. So the very first step is, of course, preparing your samples. So you will do it in the glass vial right here. And then second step is to prepare a TLC plate. So this is what a TLC plate looks like. Then the third step is to load your samples so from this glass vial onto the TLC plate just by spotting them. And then later you put the TLC plate into a capped jar and let the solvent climb up, so developing TLC plate. Then next you remove it from the glass jar and you should be able to see all these pigment spots, and by circling them with a pencil and then later try to identify what those components are. So next we'll talk about all those steps in details more. To prepare your leaves, you need to weigh out some magnesium sulfate and some sand in the paper weighing cup, and transfer your leaf with them into a glass vial and add some acetone to it and grind everything with a glass stirring rod. So after grinding, you'll let the mixture to sit for about 10 minutes. So you would do the same thing for one spinach standards and two different leaf samples. And to prepare the solvent chambers, the solvent we're using here is 4 milliliter of acetone mixing with 6 milliliter of hexane. So we measure those out in a graduate cylinder and then transfer the solvent mixture into a glass jar and then just cap it and allow the solvent vapor to saturate the chamber for a little bit. The TLC plates are stored in a desiccator just to keep them dry. So when you take them out of the desiccator, first to check if the size will fit into your glass chamber. If not, you can always trim them with a pair of scissors. Then measure about 1 centimeter from the bottom of the plate. You want to gently mark the baseline with a pencil. So remember gently because you don't want to scrape off the silica gel coating. And then make 5 light hash marks along the baseline, again gently with a pencil. Also check to make sure you are using the right side of the TLC plate. So the shining side is actually the side without the coating. So do not use the shining side of the plate. To load the TLC plate, we're using capillary tubes. So capillary tubes are these super thin glass tubes. So be careful with them because they can break pretty easily. So when you dip them into the supernatant of the leaf sample, the tube will automatically draw up some portion of liquid in there. But try to avoid the solids at the bottom because you don't want to draw up the solid. And then to load the sample from the vial onto the TLC plate, all you need to do is gently tap onto the baseline. So we made 5 hash marks, so there should be 5 spots on there in the order of spinach sample 1, spinach sample 2, and then spinach. Next, carefully place the TLC plate into the solvent chamber and cap the jar. What will then happen is the solvent will move up with the different pigments and separate them. So this is a picture of what it looks like halfway up the plate. There's actually a pretty cool video that's recording the process that you can check out on the blackboard. When the solvent is more than halfway up the plate, it is ready to be removed from the jar. So quickly do it with a forceps, and then you want to quickly trace the solvent line with a pencil. What's convenient about these pigment components is that they have colors, so you can see them clearly with eye. So circle those spots with a pencil. But sometimes there are some components that have no color, but when you place them under the UV light, they will show up. So circle those spots as well. Again, these are the pictures showing the separation process. So sometimes you might run into a problem like your spots are too big. It means your spots are probably too concentrated, and problems like if the solvent covers your spots when you put the plate into the chamber, or for the process you don't watch it carefully, the solvent will run over the end of the plate. So all these situations will affect your results. So the solution to these problems is to redo the separation process. So be very careful, try to avoid these situations from happening. A very important data analysis for this lab is to calculate the RF values for the different components. So the formula we're using here is the distance of compound moved divided by the distance of the solvent from moved. So for example, we have a TLC plate here. The distance of the solvent from moved is 5.8 centimeters, measuring from the baseline to the solvent front line. And for example, if we want to calculate the RF value for the yellow spot here, we need to measure the distance of the yellow spot traveled, which is 1.5 centimeter. Once we have those two numbers using the formula 1.5 centimeter divided by 5.8 centimeter, we calculate the RF value for the yellow spot, which is 0.26. So you can do the same thing for the blue, the green, and the red spots to calculate their RF values. And the important lab data for this lab, if you're doing this lab, you should write down the observations, like the description of the samples, mass of the spinach, your spotting plan, composition of the mobile phase, and then TLC data for both the spinach standards and the 2 samples. And for the data analysis, results, and discussion part, you need to sketch the plates with the spots and then calculate the RF values of the components of the spinach standard and the 2 samples. and then try to identify the components based on the comparison to the spinach standards. So not just the order of the spots, but also the actual RF values of those. And then answer the discussion questions. So these are the parts that you would complete in your report form. And the last slide, cleanup information if you want to read. So this is a quick introduction of Lab 8. So if you need more detailed information, you should definitely read the lab handout and other materials that are posted on the Blackboard. Have fun doing the pre-lab and lab report that are due next Monday, and thank you for watching.